Hello, and welcome back to The Vitals, the Mount Sinai Health System's groundbreaking roundtable video podcast. I'm your host, Leslie Schlachter, a neurosurgery physician assistant here at the Mount Sinai Hospital. On this episode, we delve into the most sweeping and far-reaching neuroscience studies in the history of medicine, the Living Brain Project. We're discussing all the major breakthroughs and what we know about brain science and what these discoveries mean for you, the patient. To walk us through it, we're joined by Mount Sinai doctors, psychiatrist Dr. Alex Charney and neurosurgeon Dr. Brian Kopell, who are experts in the field of neuroscience and are helping to lead this new project. Welcome, gentlemen. So as I said in your intros, you are a psychiatrist, you are a neurosurgeon.
I am.
But what is the difference between neurology and psychiatry, since there is so much crossover?
The way I look at it, I don't see a distinction between neurology and psychiatry. In fact, I think the distinction between the two is a vestige of sort of some anachronistic sort of thinking about the two entities. At the end of the day, it's all it's all brain disorders by the end of the day, and every sort of emergent behavior that comes from the brain is under the grand rubric of neurology and neuroscience, right? I mean, in essence, you are a limbic neurologist, right? In that regards, right? Your neurology happens to focus on what we, colloquially call the emotional circuitry or the limbic circuitry of the brain.
Yeah, I think the distinctions are artificial. They have historical explanations as to why psychiatry and neurology exist as two separate clinical disciplines. On the science side, there isn't that distinction. So people who train as like PhDs in neuroscience, for example, don't specialize in disorders that are classically considered psychiatric versus neurological. So like when I did my training in the genetics of brain disorders, it wasn't like the methods you use to understand the genetics of the brain differ for schizophrenia versus like multiple sclerosis. Right. It's the same principles. So on the science side, there isn't this distinction. It's all in the brain.
Or like in a book, right? Because like psychiatric diagnoses are like literally in a book,
right.
The DSM.
Right.
And there's not, like generally neurologists, there is crossover in how they treat them, but like there's a book for psychiatric illnesses versus neurologic, if we want to make it black and white.
There is, although the origins for that system are, I think sometimes psychiatry gets this rap of like, we have this very subjective diagnostic system because there is this book. But in reality, other medical specialties are just as subjective, and the criteria for diagnosis, they just don't formalize it in a book in the way psychiatry does.
Disorders very much is about the neurological exam, which is inherently subjective, right, by definition. And so there is that sort of kind of similar analogy, right?
Absolutely.
Right. So, I mean, I feel bad asking, like, what do we know now about neurology and neuroscience now that we didn't know 20 years ago? Like, what are the biggest game changers? But, like, let's just, like, psychiatry, we have to throw that in there.
Sure.
So what would you say are kind of, like, the most shocking things that we know now that we didn't 20 years ago? And, like, how did we learn to know that?
Yeah, I could take that one from, wearing my hat as someone who focuses on the biological underpinnings of these conditions, like neurological and psychiatric conditions. The main thing we've learned over the last 20 years is about how the genetics of these conditions passes from one generation to the next. If you're looking at the field 20, 30 years ago, there was this what you could think of as like single gene, single disorder mindset. This particular gene is why you are this way. Even just colloquially, we still talk like that. Like, I have the gene for being crazy, in my family or something like that. What we know now is that the genetic architecture, we call it, of these conditions is highly complex. There is no single gene for, say, a condition like schizophrenia or depression or Parkinson's disease or Alzheimer's disease. There are, in individual families, there could be a single gene that is the main cause, but across the whole population, these conditions are highly what we call polygenic. There's areas throughout your genome that are each contributing to your risk a little bit. Collectively, they all add up to increase your risk a lot, but in ways that we still don't quite understand. But that basic notion of polygenicity and the complexity of these heritable conditions is, in my opinion, the main thing we've learned over the last 20 years with respect to like their root cause.
Like, what disease states would you say has that knowledge affected the most regarding like diagnosis and treatment?
The knowledge hasn't really had clinical translation yet. So, I would say conditions on the psychiatric side, there is no condition that really does not fall into that category of our understanding of its root cause at the genetic level hasn't changed over the last 20 years. That's not really true for any psychiatric disease because genetics has changed our understanding more or less of all of them. But it hasn't changed how we treat them. Yeah,
okay.
So that translational gap is critical, and you probably make the same argument for the disorders across neurology.
Building on that, in a general sense, what has happened in the last 20 years in neuroscience, et cetera, sort of parallels, other aspects of our life. Is just this explosion of data, right? 20 plus years ago, it was a heroic effort to sequence certain genes. Now, we are able to do that at scale. That's a massive amount of increased information. Same thing with imaging of the brain, right? We used to only be able to sort of image the brain from an aspect of gray versus white matter, right? The cells versus the white matter of the brain. And now with, tractographic analysis, with resting state connectivity, we can have an entirely new sort of dimension of how we can sort of look at the brain, right? And so what the sum total of all this is I think what we're beginning to see is a challenge of this sort of 19th and 20th century sort of definition of disease, right? We think of like Parkinson's disease or OCD or depression as we learn in medical schools, these are separate diseases, right? Like with giant walls around them and never shall thee pass between the two. But as we are able to look at more and more of this data, what we're beginning to see is something like Parkinson's and something like depression has this incredible overlap because of their basic underpinnings, and that these separate diseases are in fact not. It's like a continuum. And the idea of being able to look at these entities as a continuum, I think, is a really game-changing way that we're going to develop therapies in the future.
So we're here today because we're talking about the Living Brain Project. And I learned about this a couple years ago. I'm the physician assistant who works with Dr. Joshua Betterson, and I thought, Living Brain Project, what does that mean? It means you're actually taking pieces of people's brain to learn from them. Can you, what does this project look like? How did this come to be, and what are your goals?
You want to take that one? Or
That's a big one.
I'll take it from my perspective.
Right.
Brian and I probably have interesting but related perspectives of how it started. For me, it started when I was a trainee who was coming into this field of studying the biology of brain conditions, specifically schizophrenia. That's been my focus throughout my career. And learning that the main way we were studying how our brain works at the biological level, and this is probably 10, 12 years ago, something like that, was through studies of brain tissue from people who've passed away. Right. Which, just as a clinician, felt like to me intuitively off. How am I supposed to understand the basis of people who are hallucinating, hearing voices that aren't there, or having delusions, these false fixed beliefs? How am I supposed to study the biological mechanisms of that in someone whose brain is no longer functioning, like who's passed away? So it just got me thinking of why don't we study the biology of the brain in people who are alive? And was just kind of met with skepticism from my colleagues in the field. Like well,
who's going to sign up to have a piece of their brain removed?
Or how about the almost silly notion that there was controversy about the idea that there's a difference between a living brain and a dead brain? I mean,
yeah, that's crazy.
Only in academics can you have a knockdown, drag-out fight about something that a 7-year-old would be like, look at you, like, of course there's a difference between a living and a dead brain. And, you know, I like diving head forward into these kind of controversies, right? Mm-hmm. Because that's where the interesting stuff is, right?
Absolutely. And another thing that people said at that time and still say today is that, well, if your interest is in disease X, say depression, schizophrenia, whatever it is, well, the people who get neurosurgery, which are the only people whose brain you can theoretically even consider studying when they're alive, well, they're getting neurosurgery because something's wrong. Right. So you can't learn something about how the brain works normally or how the brain goes awry in your condition of interest if you're studying people who are getting brain surgery for some other reason.
Right.
And I think that's a fundamentally flawed notion. And also when you think about what they're missing when they say that, because they're implying when you study someone who's passed away, because you could get the brain of someone who had depression or schizophrenia when they're alive, you can theoretically get brain tissue from those people when they pass away. So therefore, you can study those diseases. Mm-hmm. It's ignoring the fact that they have the worst brain problem,
right? The
brain problem where nothing works, your brain goes completely awry, which is death,
right?
And that's something that is ignored in the field.
You access the brain regularly,
right?
Can you just tell our audience, because what you do is so incredible. I've seen it with my own eyes, and I've watched you listen. Can you just explain what you do and why you do it so that we know how you guys came together?
Right. So my sort of one of the main things that I do in my neurosurgical career is a type of procedure called deep brain stimulation, which in sort of the most colloquial terms is a pacemaker for the brain. And in this sort of pacemaker analogy, we have an electrode that goes into the brain, into a specific circuit of the brain, and then it's attached to a pacemaker unit that drives electricity into the brain and modulates the electrical activity, and we, as a result, help people. So part and parcel of this approach is you have to gain access to the brain. Mm-hmm. Which involves drilling a hole in the skull.
But first, you need to get the patient in your door and get them to agree to let you do this.
Of course.
That's another
right.
conversation.
That's a separate thing. But let's assume all that has occurred. And so how do you put an electrode into the brain? Well, you have to drill a hole, right? Mm-hmm. Yep. And so the hole gives you a certain access to a piece of exposed brain. And the reason why we approach the brain in a particular way is because we know after decades and decades of intervening with the brain that there are avenues of access to the brain that are in the sort of neurosurgical sense safe, meaning you can traverse them with essentially very little impact in a patient's neurological functioning.
Eloquent versus not eloquent.
Right. Yeah, that's the sort of the old school way of talking about it. And, of course, people can like dance on a pin and basically say, well, what is eloquent? What is not eloquent? Right. My whole brain is eloquent, blah, blah, blah. Okay. But there are avenues, safe traversing avenues that you can access the brain. And we do that with deep brain stimulation. So the implication is when before we put the electrode in, you have to prepare the surface of this little brain area so that the electrode goes in with as little bleeding as possible. Mm-hmm. And, the brain is covered by a covering called the pia arachnoid, which can be tough in certain people, and it can deflect the electrode. So you want to make sure that is taken care of because, again, deep brain stimulation is like real estate. It's all about location, right. Location, right? And so if you're off target, you don't get the results you want. So the idea being is that in order to prepare the surface, I typically, and others do as well, it's pretty common in the neurosurgical approach, is you take electric tweezers and you kind of cauterize a little tiny portion of the brain. Well, when you cauterize it, you cook the brain, and so you render that part of the brain essentially inert. Right. This gives us an opportunity in certain patients, if they agree to do so, to take a little grain of rice piece of that brain prior to placing the electrode and prior to coagulating the brain. And so this gives us this really unique opportunity to gain a sample of living brain that would essentially have no impact on a patient's otherwise well-being, but can potentially move the needle seriously or move the ball down the field in neuroscience.
All right. So for what I do, and people who have watched before know I'm a neurosurgery PA, it sounds so crazy to me. You're talking about like a grain of rice level. But for us, we will literally take a whole hunk of the frontal lobe to remove a meningioma, or like a big chunk of cerebellum if we have a huge posterior fossa surgery. Are we talking like just cerebrum is helpful to you? Or cerebellum also helpful to you? Because I feel like we could be sending you guys hundreds of patients at this point.
This is a really important point, and this is how I think the Living Brain Project model scales and becomes a paradigm shift in the field, because sampling the prefrontal cortex of DBS patients is an incredible achievement. Is just a really practical insight that Brian had all those years ago. That's now we've collected over, I think, approaching 1,000 biopsies. I don't know the exact number, but by far the largest.
You need to update your website. It says 300.
Yeah, it's way more than that. So it's definitely like the largest collection of like human brain tissue for research from living people that I know of. But how do you scale this and make the general principle of leveraging access to the brain in the neurosurgical operating room as a way to advance neuroscience?
Right.
And that's through doing what you're describing, which is taking a survey of the whole neurosurgical field and saying where are opportunities for tissue collection and banking that happen all the time that we're just not taking advantage of.
Yeah, I mean, I can imagine, like, there's cases that we do, like, big skull-based tumors where I know I can think of hundreds of patients over the years that would have been like, sure, take it. But then there's, like, GBM patients where, like, it's, they're fighting for, like, every little piece of their brain to be spared. But, all right, we're getting off topic. So you guys, you are, like, what's happening at the brain level? Because before Living Brain, you're basically talking to patients, examining them, getting scans, labs, genes. Now you actually have tissue.
And more, and even more importantly, so you just described different lenses into brain function, into living brain function. But typically, in order to sort of do that, you have different studies, like one imaging study on a lot of individual brains, or you had, a lot of genetic studies on a lot of dead brains, or a lot of studies on EEG.
Right.
What's really cool about the Living Brain Project, and what really resonated with me, is that all of these different lenses are converging on a single individual brain. So we're not having to make this sort of inherent meta-analysis of synthesizing individuals from 10,000 people of different data sets and kind of creating this sort of meta sort of model of how the brain functions. We now are able to take all of these lenses in a single human brain. So
what's the question you're asking? Like, how did you get this approved?
Okay, so we got it approved by the concept is that what's the risk for the patient? That's all any regulatory agency cares about. And in this case, because the patient is losing the tissue through the cauterization process, the risk is not the right. People pick apart the word you use and how you describe the risk. It's not that it's non-trivial, but intuitively, the patient's going to lose more or less the same amount of tissue in the two options, with or without the biopsy. So the risk should be pretty minimal. So that's essentially how you get approved for the overall collection of the tissue. And then what's the question you ask? Well, with this framework in place, it's one of those things where it's not a single question study. It's more of like a framework for studying the brain. And then under that framework, you could ask many questions. I'll give you an example of a really intuitive thing that this design allows you to do that we couldn't do before. You think of like taking your blood and like measuring something in your blood that would tell you like what's going on in your brain. Like we call these things biomarkers. Like the general notion that you could use a blood sample to like diagnose something going on in the brain or predict something that's going to happen in the brain. I mean, this is something that's all over the place. You see like, some new test for Alzheimer's is out there. It's like it's measuring something in the blood that's saying what's going on in the brain. If you think about, well, what would you need to do in order to know how you can use a blood sample to say what's going on in someone's brain? Well, just what would be the right study design to even create that knowledge base? Well, you would want to have blood from someone and their brain at the same time.
Right.
And you'd want to do that in a lot of people. And you want to profile the blood and profile the brain, then look at how is what you see in the blood related to what you see in the brain. Well, that study's never been done. So, like, everything we know about trying to, like, use the blood to, like, measure what's going on in the brain is not based on the correct study design.
Yeah. So we don't even know what we are going to know. Well, you guys might know now.
We're learning.
We're just beginning.
Yeah.
I mean,
what have you learned so far?
So I'd say the key things we've learned from the Living Brain Project, number one is that you can collect brain tissue from living people in a safe way. That's a key.
We just published that in like over 12 or 1,300 implantations, and we looked at they were roughly half and half, and we looked at all of the safety data that we typically look at in a deep brain stimulation operation, anything from cognition to bleeding to seizure to infection, and there just is no difference whether I took a biopsy or not.
So you're looking like obviously retrospectively to the patients that you didn't versus the patients that you are. It's about the same.
Yes.
Okay.
Yes. And so as Alex points out, we can do this safely at scale. That's
when we first proposed the study.
Like the biggest thing.
The first thing people said was, "You can't do that." Well, you can do it. It's safe. And I
remember hearing those conversations, and I was like, "Guys, we're talking about the smallest little bit."
Yeah,
they're there anyway.
Right. People said that weren't neurosurgeons.
Yes,
right.
or it depends on the type of neurosurgeon. But like most bread and butter neurosurgeons, if you were to ask them, "What do you think the effect is of taking a tiny piece of prefrontal lobe of the brain on terms of someone's function?" they would probably laugh.
They'd laugh. Yeah.
They would laugh at first, just in a way that only a neurosurgeon could laugh to that kind of question.
You'd have to be in a neurosurgical OR to be like,
of course,
of course you can,
right? And so, but of course, there's always opportunity for controversy, in anything that we do, basically.
Yeah. The second thing we've learned is that the living and postmortem brains are not the same at the molecular level.
Your
so
7-year-old was correct. Basically,
what's the biggest thing that you've learned so far?
If you measure the types of things we measure in these brain samples are gene expression levels, so we call it transcriptome-wide studies. We measure the expression level of every gene you can detect or every protein. So you're talking testing tens of thousands of genes, tens of thousands of proteins. So more or less, it seems like there's statistically significant differences between the living and postmortem state with respect to like every gene and every protein.
What are you doing with this piece of tissue? Like, is it just like one piece going on one slide, multiple? Like, how are you fixing it, and how are you because you want to have a database to look for it in the future. So how do you
the general principle when we take a biopsy is to keep it in as pristine condition as possible, to leave open the opportunity to do as many things down the road with it as possible.
Right.
Depending on what you want to do with it, you may have to process a specific way at the point of collection, which then oftentimes requires you've essentially committed to a specific path of only doing that particular test on that sample. Mm-hmm. So we try and process the sample in a way at the point of collection that leaves open the most possible things we can do with it down the road. So for a given biopsy from surgery to surgery, we may not be using that tissue for the same exact set of things. So, for instance, one interesting thing that we published recently in this journal, Nature Biotechnology, this is work led by our colleague Yizhou Dong. He reached out to us in learning about the Living Brain Project, asked us if we could spare 3 or 4 biopsies because he had developed a mechanism to deliver gene therapies to the brain in mice, and he wanted to verify that when treating human brain tissue with that, it's essentially like a new kind of pharmacology, mm-hmm. That he can show that this delivery system delivers genes to the human brain.
Wow.
So you need living human brain tissue to do this, right? Because it's you're essentially treating the tissue.
How long does it stay alive for?
Yeah, it's a good question. And the answer is it depends on how you process it. If you mm-hmm. get it right from the OR to the lab, you can keep the tissue functional for assays like this for an hour or two. But we haven't systematically.
You made that sound like that was a long time, but I'm like, that's not very long.
No, it's not very long. Yeah, but it's long enough to do something, and you could probably keep it alive longer if you came up with creative ways.
Right. So there's an opportunity to keep it, to learn how to figure out how to keep it alive longer.
Yeah.
Right.
So for people listening, this is like a really big deal. Like, you guys have moved the needle in research. That, like, is anybody else doing anything like this?
There are other groups that have studied. Well, I'd say if you back up, what we're doing is not actually that novel. Like, because if it's almost like rediscovering an approach that's like as old as neurosurgery and psychiatry and neurology itself. Like, you think of like Wilder Penfield and like the homunculus that we all have to like learn, right? Like, it's like this notion of how the brain is organized and what areas of the brain do what. These came from innovative studies performed in the operating room, like bringing together neurosurgery and neuroscience. So I actually think what we're doing is essentially like the same philosophy. It's just like updated for like using like state-of-the-art technologies like genome sequencing and whatnot.
I guess it's like what you were talking about at the beginning is like all of this information that you've gotten over the last 20 years. You can actually now use it. That you're getting living brain, right?
Right.
Okay.
And the tools that we have to sort of deal with these growing high dimension data sets is getting better. Like, we have more tools today to deal with more information than our colleagues from decades ago, right? Mm-hmm. We have computers, we have now machine learning tools and things of that nature that we can now begin to take these incredible data sets and do something even more interesting from it. So the novelty very often comes from revisiting the old and doing it in a certain and sort of in a new way, basically, right? Because like you look in the Bible and King Solomon said there is no new thing under the sun. So there's this notion that goes back to antiquity that novelty is a very funny cyclical thing,
right?
Mm-hmm. Right, and I think the same thing is here as well. We're doing something that is very old in a very new way.
Yeah, it's the whole notion that the people who I was being exposed to when I started the Living Brain Project with Brian, the people who are doing the work on the genomic side and trying to understand the biology of these conditions, that they were not working at all with neurosurgeons. I mean, when you think about that, that's crazy. How could the world's leaders in studying the biology of the human brain not be working hand in hand with the people who hold the human brain in their hands and living people, and no one was doing it. And right, so it just seemed like if you really want to understand how our brains work, neurosurgeons and the people studying the biology of the human brain need to be working like hand in hand,
right?
And they weren't,
right? Like, and that goes back to the original question you asked, like, how did this all come together? And for me, just personally, the thing that resonated with me growing up as a kid was, the Apollo program. And what was the essence of the Apollo program? It was the coming together of different disciplines to pull in a certain direction to make a miracle happen. We landed on the moon.
So what's our miracle here? Like, like, I know you guys, you're gathering all this information, but for like patients listening and for me who like, I understand everything you're saying, I want to know, how is that going to help me or my child?
So when I think of it, the way I explain it to people is you think about, the course of your day, all the things you feel and experience. It's like you feel sad or you feel happy or you drink some coffee and you got a pep in your step or you feel bored listening to a podcast, someone talking about the brain. Like over the course of your day, your brain is doing all these incredible things, and all of that ties into to mental illness and neurological illness, all of those brain functions. And if you asked, well, what's happening inside my brain, like as I'm experiencing those things, like when my mood goes down all of a sudden because I just learned that something bad happened, like I lost a loved one, like what happened inside my brain? This is the study that needs to be done to answer those questions at the most basic level. It hasn't been done before. We have the technology to do it now. So like that's the moonshot, is really being able to make this map between how our brain is functioning at the deepest biological level, and then our subjective experience as human beings. Making that connection is what the Living Brain Project's all about.
If you were to guess a timeline on when there would be, like, not earth-shattering, but just like, wow, helpful information, when do you think that would be?
Well, I would argue that Alex has already given an example of it, the development of a new tool to treat patients with Alzheimer's.
Right.
So I'm just saying we're really just scratching the surface.
Wait, to be clear, you mean the development of tool, like, so that blood test...
So I think the most, if I had to say, the most practical thing for patients that the Living Brain Project has led to today would be that we have used the tissue we collect in this way to show that a new class of drugs, which can be thought of as like the next generation of medicine, that they work, they get into the brain tissue of people. And
what drugs are we talking about?
They're called gene therapies.
Okay.
So these are drugs that you can deliver through an intravenous injection.
Okay.
And they allow you the drugs go into the brain, and you can now modulate essentially any gene.
Okay.
Right. In the past, you
Very
think
cool.
of pharmacology, you've got a couple hundred genes that you could target through all of the drugs that exist in the world that are like approved for use in humans right now. This technology opens up the druggable genome, what we call it, to every gene in our body.
So let me give you another.
There's like so many things adding up in my head. This is painful.
So another study that we recently published, and it's one of my sort of one of my favorite things was when we asked ourselves the question. So during the course of a deep brain stimulation procedure, I record neural activity deep in the brain near the target of
these are the
where
beta
these
waves?
electrodes. They can be beta waves, but we're actually recording single neuron recordings. So beta waves are the confluence of many neurons working together. One of the actual signals that we record during deep brain stimulation is single unit recording. So just a single neuron. We get a lot of electrophysiological signals from multiple single neurons. And so we sort of just asked the question, is there something that we can correlate and we can connect of the expression of the genes at the surface with these neurons that are, for all intents and purposes, a universe away? Because again, the distance is in the brain. The brain is all interconnected. So even though the distance is large, things work together. And the idea that there may be some genetic sort of link between that and the was sort of like, well, maybe, but it's hard. There's not a direct connection. So it was like, well, it seemed like a long shot, if you will. And lo and behold, what we found is, in fact, there is a connection. And so why is that interesting? Well, it's interesting if you're a neuroscientist just from the notion of it. But here's something that you want to talk to your lay audience, like, how is this going to affect me? Well, as a neurosurgeon that's interested in putting devices in the brain, people now have all heard about brain-computer interfaces, right? And so right now these devices are relatively dumb. They're just electrodes that are placed on the brain, and then we have to deal with the information. But you could imagine the data that we are collecting in this type of process could create a data set to teach a brain-machine interface to work really well. Maybe you want, right now we conceive of brain-machine interfaces as just affecting motor function, moving, right? But in principle, this kind of data with electricity, we could potentially influence gene expression if done in a sort of granular way. I mean, again, I am talking in a sort of loose way, but that's what the potential is for this type of study.
I'm thinking about, like, I've done so many podcasts now on issues that, like, autism, for example. Like, when I feel bad, I'm blanking on his name, but he was saying the biggest thing that we've learned about autism is it's genes. There's a spectrum. And I would gather that if you were to get, parents of an autistic child, teen or whatever, and you said, hey, there's something that we could we can give them.
Mm-hmm.
That's
what we do.
That's amazing.
Yeah,
That's a big deal.
So we have a program now that does that. It's built off of discoveries that were made through the Live and Brain Project. So because we were able to, like, I don't know how much the audience may really understand this immense challenge you have when you have made a discovery related to like a new drug in a mouse, and how hard it is then to translate that information to a human being.
Right.
Where like almost every single drug that gets developed and then first treated in an animal never makes it to even be tested in humans, or if it is tested, never makes it to actually get approved by being effective.
Right.
So the ability now to use the living human brain samples as this new step to verify that the drug you've developed does influence human brain biology, that's a critical step.
Oh, yeah.
Yep. Now you've got a new level of evidence to, like, when you're applying to the FDA to, like, look, this drug that we're proposing to use, it doesn't just work in animals. It actually gets into the human tissue and has the intended effect. So this opens up the ability to do things like test and then ultimately bring new drugs to patients with conditions like autism. And we're actually doing that specifically for autism and schizophrenia right now.
Outside of like podcasts and things like this, how do you teach the general public that, like, this is how you move the needle and how we actually eventually can treat these illnesses?
We collect the data, we collaborate, and we publish, and that's the best way.
You hope it trickles down.
Yeah.
You know, people these days do TikToks for stuff like this.
Yeah,
so you know.
I think probably me and Alex are not that this doesn't come naturally to us to do this sort of thing. So, right? I mean,
unfortunately,
right? We are, sort of not of that bent. But yeah, you're right. I mean, there's new ways to communicate to people. It's a thought to consider.
Yeah.
For sure. But the collaboration goes on. I mean, that. To me, that's the most important thing. That's my North Star. It's sort of, I assume it's Alex's as well. And we continue the work.
What advice would you give to early researchers, people just starting out their career that want to follow paths similar to yours?
The advice I give is to challenge the status quo.
Mm-hmm. I was literally going to say the same thing. And ideally find a partner or colleagues that are of that same mindset, and hopefully in different disciplines than your own, so that the amalgam of your disciplines are greater than the whole. And that's what I recommend.
I think a true test of the Living Brain Project's impact will be, if there's some other young researcher who hears about what we do and thinks, I could see a way to do it better.
Right. Mm-hmm. I agree.
Yeah.
Yep.
One of the things that frustrates me a lot about what you do is the percentage of patients that actually get the care that they need from you. So for us, we take care of patients that have tumors and aneurysms, and they're literally running. They can't wait for their appointment to come in, and they want the tumor out of their head. They want their aneurysm clipped. But a statistic that I don't love for what you do is only about 3% of patients that need your treatment are actually getting it.
Yeah.
So how do we get them in the door to you and then to participate in a project like this?
It's the notion, in surgical procedures, there's this sort of definition between elective and emergent.
Right.
And emergent is like anything that's going to kill you right this second, and there is no debate about it. You either do this operation right at this moment, or you're going to lose life or limb, right?
Yeah.
And then there's everything else is called elective.
Yeah.
But the elective stuff, like taking a brain tumor out in a planned way, is considered
elective.
It's elective. But
yeah.
Is it really? In other words, who's going to elect to literally keep their brain tumor, right?
So many people actually elect to keep their brain tumor.
Right, but like you understand what I'm saying. In other words,
I do.
And so the stuff that I do is almost truly in the essence of elective, in the sense that there are so many other treatments that these patients are presented with that the notion of undergoing neurosurgery becomes a much more daunting issue. And it's only daunting because we have failed as a field to really sort of express the grand improvements in quality of life of this approach to just medications alone. And that's not a failure of the patient's understanding. I would put that failure on us as a field that we have
like
yet.
The treating neurologists need to really know.
And I mean the field, I mean neurosurgeons,
right.
neurologists, psychiatrists, all of us together have not been able to truly communicate the impact of quality of life on this type of procedure on patients' lives. Okay. And, we just got to do better.
Yeah. To be really clear about what he's saying and add in diagnoses.
Yeah.
Dr. Kopell is a neurosurgeon who specializes in a treatment called deep brain stimulation, DBS. And historically and still, there's patients out there who have Parkinson's. And in Parkinson's, you can have a tremor. For many patients, they're treated by their neurologist. There's medications. Some don't even get the appropriate medications. But if people have such bad tremor that it's affecting their life, meaning they can't even pick up a pen or give themselves water, there's a procedure that they can do called deep brain stimulation, where neurosurgically electrodes are put into the brain and stimulate a part of the brain that calms the tremor, either cures it or diminishes it enough that they have improvement in their life. And so the statistics are there's only about 3% of patients out there that are actually getting this treatment that could benefit from it, which is a crazy number. Like that's 97% failure. So if you know someone that has Parkinson's, they don't even need to even have that bad of a tremor yet.
Right.
Like to get into doctors that know how to take them down the path. And tremor from Parkinson's is just one thing.
Yeah, I mean, the truth is like, the mainstay of Parkinson's therapy. I'm going to say something pretty controversial. I'll probably get yelled at for this,
Let's
but
do
whatever. Let's do it. So the mainstay of Parkinson's treatment is the replenishment of dopamine. Right, that's the fundamental original.
We can
sin
all agree on that.
Of Parkinson's is that your dopamine producing cells die, and so we have to give back dopamine. Well, the truth is, we do that by giving either pills or pumps or so on, but the way that the brain organizes its own dopamine use is very, very specifically at contextually appropriate moments. We don't have the ability to do that, so we just give you a giant wave of dopamine that you're just getting on a daily basis. Mm-hmm. And early on in the disease, that's fine. But, more and more that we're beginning to see is that over the long period of time when you mess with the dopamine system, and we all know this based on social media and things of that nature, is that your dopamine system gets dysregulated. And that is the essence of the quality of your life, right? Dopamine is our reward mechanism. It's what allows us to pursue difficult things. And if that becomes deranged, then your quality of life is decimated.
Right.
One of the great things about deep brain stimulation is that it allows us to sort of mitigate that wave of dopamine replacement and still maintain your motoric benefit so that you can move and do your life and have all the quality of life. But we're minimizing this aphysiologic, replenishment of dopamine. Mm-hmm. And that has an implication in terms of your quality of life. One more thing as well. What happens when patients get advanced Parkinson's disease? They stop exercising. They stop eating protein because the protein interferes with their medication. So you begin to sort of do things in your life that actually hurt your longevity.
Right.
If we can intervene DBS much earlier, then we can avoid those behaviors that really are going to actually impact your longevity.
And maybe future gene therapy.
There you
Definitely.
go. So we talk about a lot today. There's like this overlap of neurology, psychiatry, neurosurgery, neuroscience. What would you say is the biggest bit of misinformation in your field specifically? You guys will each take a turn.
Yeah, well, I'll talk specifically about people who have schizophrenia because it's an easy answer when you think about that because no one understands. In the general public has a good understanding of what it means to be schizophrenic, people walk by individuals on the street suffering from schizophrenia who are hearing voices that aren't there and talking to themselves, and it's easy to brush them off as, oh, they're just crazy or they're on drugs, and these are individuals who are suffering incredibly. So I think the general lack of understanding in the public as it relates to severe forms of mental illness. I think common forms of mental illness like depression, anxiety, the public has gained a lot more sensitivity to over in recent years, but still the more severe forms like psychosis still are these individuals are ostracized and poorly understood. And that's something that I think a lot about, because that's the patients that I treat and focus on in my research.
The biggest misinformation is something we already just touched on, the idea that this deep brain stimulation is a dangerous procedure, in the sense that I think that the danger and the risk of this procedure is so overblown, and patients, of course, are going to sort of make that overblown because they're lay people, but I think that it's not based on reality. And in fact, there was recently a paper that was published that looked at the rates of morbidity complications compared to like a lot of commonly indexed procedures like C-section, for instance. Right. And deep brain stimulation is way safer than these procedures that we typically think of. Oh, it's no big deal. Oh, it's a common procedure. One of the really nice things about deep brain stimulation, and the fact that we've been doing it now for 30 years, is that we do it really safely. And to leave quality of life on the table in the face of that relatively safe procedure, I think is a huge piece of misinformation. And if we can overcome that, we'll help a lot of people. Yeah,
because it's not like for the tremor, it's not just the tremor that you're getting rid of. It's the ability to eat, feed themselves, of course, exercise, everything. It's all included.
All of the downstream effects that we as a field of medicine are sometimes, it's hard for us to convey that point.
Right. I have a similar situation in my practice. We treat a lot of patients that have trigeminal neuralgia, severe facial pain, and they come to us after they've been on, months or years of medications, multiple medications. Their pain is refractory, and it's, I guess, there's like kidney stones, trigeminal neuralgia, and then like what's the chronic regional pain syndrome? There's
CRPS.
Yeah. By the time they come to us, they're like, begging for us to drill a hole in their head to do the surgery. And I think even then we only treat like 20% of people that could benefit from it. So to lay people, drilling a hole in your head to fix a problem may sound crazy, but it's actually proven decades to be safe and help people. So I will volunteer to participate in the Living Brain Project if there's a way that you can learn of what happens in my cycle when I before, during, and after, what those hormonal changes are. I'd love more information on that. So would every woman ever. Is there anything that you guys want to add? I feel like we just touched on the tip of the iceberg. I'm very interested in this, and I hope people got the point from this today. But is there anything else that you guys want to add that we did not discuss?
No, thank you for the opportunity to talk about it.
Yeah, it was good.
Great. Thank you guys so much for being here. I appreciate your time.
Thank you.
You bet.
So that's all for this episode of The Vitals. I'm your host, Leslie Schlachter. Subscribe to The Vitals and the Mount Sinai Health System's other video podcast programming on YouTube, Apple Podcasts, Spotify, or wherever you get your podcasts. To learn more about Mount Sinai's Living Brain Project, or to book an appointment with a Mount Sinai psychiatrist, neurologist, or neurosurgeon, scan the QR code on your screen or click the link in the description below. To get in touch with the show or to suggest an idea for a future episode, email us at podcasts@mountsinai.org.